All right, guys. So, like I said, today's topic, new topic, is how are our genes regulated? Key thing, we know that in every cell in your body, the DNA is exactly the same, right? Except for sperm and eggs. But all your cells look the same way. No. That's because of what we call selective gene expression. Think about what that means. Certain genes are selected to be owned in your skin cells that aren't selected to be owned in your brain cells, your stomach cells, your intestine cells. Different cells have different genes that are working. Different genes that are turned off. Selective gene expression. Some genes are turned on in some places. Those same genes are turned off in other places. That's what we're talking about here. Because of that, that leads to what we call cell differentiation. That's why your skin cells are different than your stomach cells and your pancreas cells and your brain cells. Because different genes are working in those different kinds of cells. Does that make sense? You guys are trying to nap on me, hop on up and listen. So we're regulating our genes. Because we can turn on and turn off certain genes, that lets us respond to our environment. For example, if, let's say the blood glucose levels go too high, what genes do you want to turn on? Genes that make insulin. Okay? If your blood glucose levels go too low, you want to turn on the genes that make glucagon. Not glycogen, glucagon. That's close. If something's going on in your physical environment, maybe somebody beat the dog out of you. You might want to turn on the genes to make what? Adrenaline. You might want to get that fight or flight thing going on for you. Okay? This also lets us respond to hormones. And we'll talk about how the hormone thing works in a second. But here's the thing. We have these proteins in our body called gene regulatory proteins. These proteins allow us to turn on or turn off certain genes. Very important. Gene regulatory proteins. They turn off or turn on certain genes by directly bonding to the DNA. They're like a key. They turn on a gene or they turn it off. Gene regulatory proteins. And again, by turning on or turning off DNA, we call that selective gene expression. That lets our cells become different. It lets different cells do different things. Division of labor. Pretty important stuff. Now, we understand a lot more about how this works in bacteria than we do in people. This bacteria is simple. How many chromosomes in a bacteria? Just one. What's its shape? Circular. It's pretty simple stuff. So in bacteria, there are only a few genes, because one chromosome doesn't have that many genes on it, right? So that's where we started learning about genes is in bacteria. We're simpler. We know today that bacteria have a structure called an operon. And this is really important for you to understand. An operon is a set of genes and the DNA that controls those genes. So it's a set of genes and the regulatory parts that go with that gene setup. The genes and the pieces of the DNA that help to control that particular gene, whether it's on or whether it's off. It's only in bacteria. It's only in prokaryotes. Hence the heading here, prokaryotic gene regulation. Human beings do not have operons. We do it a different way. There's lots of different stuff going on. We still don't understand it all in eukaryotes. But in bacteria, they have these structures called operons, which is a set of genes and the mechanisms that control those genes, whether they're on or whether they're off. And for the rest of the time today, mostly we're going to concentrate on what an operon is. An operon has four main parts. First part is called a promoter. And before we talk about what that promoter is, stop writing for just a second and listen to it. For a gene to be owned, what has to be happening to that gene physically? a gene is on, what has to be happening to the gene? Think central dogma of genetics with me for a minute. For a gene to be on, it has to be making what? What do genes make? Proteins. Okay? For a protein to be made, two things have to be happening to the DNA. Remember, DNA goes to what? mRNA. That process is called transcription. For a gene to be on, transcription of that DNA has to be happening. Also, that mRNA will go out to the ribosomes where it will be. mRNA will go to the ribosomes where it will be, I'm looking for a verb here, translated. Okay? And it will be used to make protein. Now, we know tRNA is involved. tRNA helps translation happen. This is the big idea. Transcription and translation have to be happening if a gene is on. So, that being said, the promoter is the part of the DNA where the RNA polymerase binds. What's RNA polymerase do? RNA polymerase. It opens the DNA up and then does what to the DNA? RNA polymerase. It makes RNA. It does which one of those processes? Transcription. It's the enzyme of transcription. So this is one of the places where the gene gets turned on. It's where the RNA polymerase binds and starts transcription. Starts making the gene work. That's why it's called the promoter, because what does it promote? Transcription. It promotes transcription. All right. Right. Now remember, we're talking about a string of DNA here. So for the first, if this was an operon right here, the first piece of that DNA is the promoter. This is like one string of DNA. After the promoter is a piece of that DNA called the operator. Think about the old-fashioned operators of phones. You guys watch the Andy Griffith show? When they made a phone call, they called the operator, and they said, I want to talk to Barney. And what did the operator have to do? She had to physically take a wire and connect the line that Andy was calling on to Barney's line, right? Okay? She had to physically connect those lines. The operator was like the control unit, right? She was the regulatory part of that whole phone system. She connected you if she wanted you to work, okay? That's what this does. The operator is basically like the on-off switch of the operand. Notice up here it says site of repressor binding. We'll talk about what that means in a second. Yeah. We'll talk about what the site of repressor binding thing means in a minute. After that, though, is the most important part of the operand. It's the structural genes. These are the genes that we're trying to control. This stuff up here is not a gene stuff. This is the control parts. These are the genes. These are the genes. This is what we're trying to turn on or turn off. The structural genes. Somewhere else in the DNA, it may not be right after the structural genes. It may be somewhere else. But somewhere else in the DNA is another set of genes called the regulatory genes. These genes make what's called a repressor protein. They make a repressor protein. The regulatory genes code for what are called repressors, or repressor proteins. Here's the deal. Let's say RNA polymerase binds to our promoter. It's ready to do transcription and turn the gene on. But stuck right here to this operator is a big thing called the repressor protein. If RNA polymerase is over here, can it get to over here? No. If this is sticking here? No. So the repressor proteins, if they're bound to that operator, what can we say about that gene? If there's a repressor setting right here, what can we say about that gene? It's off. If there's not a repressor setting here, we can pretty much say that gene is what? On. So if a repressor is bound to the operator, our operand is off. If a repressor is not bound, our operand is on. Simple as that.
Sounds like condoms.
Kind of like condoms, yeah, similar. So let's look at a little bit more detail here of how this works. Still copying? Okay, go ahead.
Okay.
Everybody got what you need here? All right, so here's a picture, a little bit more detailed picture of what I drew up here. Here's the promoter where RNA polymerase binds. Here's the operator where there may or may not be what? There may or may not be what here attached to this operator? A repressor protein. Here are the genes we're actually trying to turn on or off, the structural genes. Somewhere else in the DNA are the genes that make the repressors. They may be before, they may be after. But right now, is this operand on or off? It's off, because attached to its operator is a repressor protein. Right now it's off. If we want to turn this thing on and start transcription, we have to get that off of there. We have to have a way to remove it. And that's kind of what we'll end the day with, is talking about how does that happen. There's two kinds of operands in bacteria: inducible and repressible. Inducible and repressible. Let's say I could induce Maddie to study. Maybe I can't do that, but if I could, what could I do? What am I doing? If I can induce Maddie to study, I'm causing her to study, right? I'm making her study. So inducible operands are operands that usually are off. Maddie's usually not studying, okay? But with the right little help, she might be convinced to start, right? So inducible operands are operands that control genes that normally the bacteria don't need. Maddie's smart enough, she doesn't need to study most of the time, right? Right?
Yeah, on the exam.
So inducible operands are usually off. They code for genes that the bacteria usually don't need. Example. The example you're going to hear always, and probably the only one they would ever ask you about in an AP exam in bacteria, is an operand called the lac operand. As soon as you see lac, you ought to be thinking what? LAC.
Milk.
Milk. Right? Milk, sugar. Lactose. Bacteria. Do they usually have lactose that they can eat? No. So does it make any sense to make lactase if you're a bacteria? Lactase is the enzyme that breaks down lactose, right? So that's what this is. That's what this operand codes for. It codes for lactase. It doesn't code for milk. It codes for enzymes to digest milk sugar. If you're a bacteria and you're not around milk, does it make sense to waste time and energy making enzymes to digest milk? No. Okay? But so most of the time these lac operands are off. There's no sense in making lactase if you don't have milk. If you don't have lactose. Okay? No sense in making lactase if you don't have lactose. So they're off. But let's say you're a little bacteria and all of a sudden you find yourself floating in a container of milk. That means there's basically an unlimited food source for you. But right now you can't what? You can't digest it. But you do have some genes that allow you to digest it. So what are you going to do to those genes? You've got to turn them on. Okay? You've got to induce them. And what is the inducer here? What is making them turn on? The presence of what? Lactose acts as an inducer. It turns these genes on. So lactose actually leads to its own demise, right? Lactose turns on the genes that end up doing what to lactose? Breaking it down, digesting it. Does that make sense? Everybody with me on this?
The bacteria starts living there and then it gets molded.
So this whole idea of an inducible operand, it's used to conserve resources. It's used to regulate the genes. Is there any sense in making stuff you don't need?
No.
No. But if all of a sudden you need it, if you're a bacteria and you find yourself in a big jar of milk, well, it makes sense. You don't start eating that milk. So you start making lactase. And therefore, drink. Bacteria don't drink, right? So they're eating it. So here's the deal. Lactose acts as an inducer. Here's the cool thing. Let's go to a picture here first. All right, right now, here's our lac operandi. It's off because there's a repressor bound to the operator, right? It's off. But all of a sudden, this bacteria finds himself in the milk. Some of that lactose will actually come in and bind to that repressor. Here's lactose bound to the repressor. When lactose bound to the repressor, what happened to the repressor? It detached. And now there's no repressor here, so what can happen? Okay, but what has to happen first? For a gene to be on, what has to happen first? Transcription, followed by translation. Okay, so the lactose is said to be an inducer. It binds to the repressor and makes it come loose. And therefore the gene is turned on. The DNA is used to make mRNA, that's transcription. The mRNA is used to make enzymes, and that is mRNA to protein is translation. Okay, so we got transcription, we got translation. We made lactase. Lactase is actually a series of enzymes. And now the bacteria eats the sugar, it gets nice and fat, makes babies, everybody's good. Okay? Well, the bacteria are lactose intolerant right now, right? And now they're not. Okay? So they go from lactose intolerant to lactose tolerant.
Well, I mean, are they just not able to?
Oh, there are some species of bacteria that would not have this operand at all, right? Right. The second kind of operand, and what we'll end with today, is called a repressible operand. Repressible operands. Repress means to stop, right? Repress. So these are operands that the bacteria almost always needs. They're operands that the bacteria need to have owned. They code for things that the bacteria need almost all the time. But occasionally, they may not need them. So what do? Turn them off. The only example I can think of that they might ask about on an AP exam about a repressible operand is something called the trip operand. Trip operand, TRP. That's an operand that bacteria use to make an amino acid, tryptophan. It actually makes the enzymes that make this enzyme. It makes the enzymes that make this amino acid. What's tryptophan kind of famous for? First of all, it's an amino acid that doesn't end in INE, right? Most of them do. What's it famous for? Tryptophan. We talked about this the other day in class. It's in Turkey. There's a lot of it in Turkey.
Like
Sleepy, yeah, kind of. Sleepy.
It'll make you sleepy. Okay? If a human being eats a lot of tryptophan, you just get sleepy. It's like a natural sleep thing. It's in turkey. It's also in milk. Makes you just want to go to sleep.
that might
But-
be hormones.
Yep. But here's the deal. Join me, guys. The trip operand is a set of genes and the things that control it that make enzymes that end up creating tryptophan for bacteria. Tryptophan is an amino acid that bacteria has to have. They gotta have it. No, just not for bacteria. But take the sleep thing out of this conversation. Tryptophan makes humans sleepy, not bacteria. Well, it's an amino acid they have to make bacteria proteins. They have to have it. And typically, they have to make their own. Yeah. It's an operand that's usually owned because it's making things that bacteria have to have. And I'll talk about why we might want to turn it off in a minute. It can be turned off, but usually it's on. So bacteria almost always need to make their own tryptophan because they're usually not eating foods that have it in it, right? So they have to make their own. But let's say, around Thanksgiving, you're a little bacteria and you're on somebody's hands. You don't usually have much tryptophan, so you have to make your own. But all of a sudden, that person picks up a big turkey. And now you get walked onto the turkey. And there is tryptophan everywhere. Okay? does it make any sense for you as a bacteria to continue to make tryptophan if it's all around you? No.
You can just eat it.
So in this case, you want to turn off that operand, right? Because you don't need to make it anymore. You repress that operand. You conserve your resources. You regulate your genes so you don't waste stuff. So this repressible operand, again, is usually on, but sometimes you turn it off if you don't need to make it. The trip operand codes for enzymes that make the amino acid tryptophan, which is hugely important for bacteria. If they don't have it, they die. And usually they don't have it, so they have to make their own. That's what the trip operand does. But if that bacteria finds itself on a piece of turkey, there's no need to make tryptophan because it's everywhere around it. It's common. So use what's there. So let's look at this picture real quick, and then we'll go from there. This is the trip operand. It's usually on. The DNA is being transcribed, then being translated. It's making the enzymes that make tryptophan. But now, oh my gosh, now this bacteria finds himself in an environment where there's lots of tryptophan. That tryptophan acts as what's called a co-repressor. It binds to the repressor, which turns that repressor on. And if the repressor is on, that means it's going to stick where? What's that place where it sticks called? The operator. And now what have we done to that gene? We turned it off. Turned it off. Because there's already tryptophan there, there's no sense in making it. We'll revisit this next time. Let me turn off the recorder here.